Imagine, just for a second, that you're tasked with finding a single specific needle hidden entirely inside a massive microscopic haystack. And not just finding it, right? But proving exactly what it looks like and exactly how much of it is actually in there. What if you could detect a single protein inside a complex mixture of tens of thousands of proteins? It sounds literally impossible. But what if I told you there's a signal so incredibly specific that it can reveal a hidden disease, confirm a gene's expression, or validate an entire biological mechanism with absolute precision?
Well, that is exactly what the Western Blot does. Welcome to this explainer. Today, we're going on a high-energy, highly structured journey through what is undeniably one of the most essential laboratory tools in modern life science. We're going to break down the core principle of Western blotting, every single crucial step of sample preparation, and the complete lab workflow, literally from a simple gel all the way to final detection. Let's enter the fascinating world of protein analysis. Now, before we get our hands dirty with the actual lab mechanics, we need to define what this molecular verification system actually achieves. A Western blot is an analytical technique used primarily to detect a specific protein of interest hidden within a complex biological sample. But, it goes way beyond just a simple yes or no detection. It allows researchers to measure relative protein expression, meaning how much of the protein is actually there, confirm its exact molecular size, and powerfully validate antibody specificity. Honestly, it's like the ultimate biological lie detector test.
So, to understand how this all comes together, here's our roadmap. 1. The core principle. 2. Preparation and separation. 3. Transferring and blocking. 4. The antibody detectives. And 5. Detection and interpretation. Let's jump in.
Section 1. The core principle. Okay, let's dive right into this. Taking a macro-level view of how this operates, a Western blot is really built on 3 sequential scientific concepts that form the backbone of the entire methodology. Step 1 is protein separation, where proteins are sorted out by their size using SDS-PAGE electrophoresis. Step 2 is the transfer, where we move those sorted proteins from a super fragile gel onto a solid membrane. And step 3 is immunodetection. This is where specific antibodies bind to our target protein and produce a signal we can actually see. Now, if the complex chemistry starts feeling a bit overwhelming at any point, just keep this brilliantly simple analogy cheat sheet in mind. The gel, it's just a sorting machine, lining proteins up by size. The membrane is simply a permanent protein snapshot, capturing that sorted lineup. And the antibody is your molecular detector, zeroing in on exactly what you're looking for. Keep these 3 concepts in your head, and I promise the rest of the chemistry will make perfect sense.
Moving right along to Section 2, Preparation and Separation.
All right, shifting our view down to the microscopic level. Before we can sort our proteins, we have to actually get them out of the cells using a precise chemical toolkit known as a lysis buffer. Now, this isn't just soapy water, right? It's a highly engineered cocktail. You've got tris HCl acting as a pH buffer and NaCl to maintain ionic strength. Then you add SDS, which denatures or unfolds the proteins, alongside detergents like Triton X100 or NP40 that violently disrupt the cell membranes. Finally, and absolutely crucially, you drop in protease inhibitors. Think of these as little molecular bodyguards that stop cellular enzymes from chewing up your precious proteins once the cell is broken apart.
So, what does this actually look like physically in the lab? Well, it's a very systematic procedure. First, you wash your collected cells or tissue with cold PBS. Then, you add that potent lysis buffer we just talked about. You let it incubate on ice for about 20 to 30 minutes, vortexing it intermittently just to shape things up. Finally, you centrifuge the whole mixture at really high speeds. We're talking around 12 to 14,000 RPM for 10 to 15 minutes at 4 degrees Celsius. The heavy cellular debris sinks right to the bottom, and the remaining liquid on top, the supernatant, is your absolute gold mine. That is your total protein extract.
But hold on a second. Before we send this extract off to our sorting machine, we have to measure our payload. This is called protein quantification. Using really common methods like the BCA or Bradford assays, we calculate the exact concentration of protein in our samples. And why does this matter? Well, because if you want to compare samples accurately, you absolutely must ensure equal loading across all your wells. You can't exactly compare the protein levels of a diseased cell to a healthy cell if you started with twice as much biological material in one of them.
Now, what's really interesting about this next step is the brilliant physics at play during the SDS-PAGE phase. Proteins normally come in all sorts of wild 3D shapes and charges, which would make a race to the finish line totally unfair. So what do?
We mix the sample with an SDS buffer, a reducing agent like beta-mercaptoethanol, and we heat the whole thing at 95 degrees Celsius for 5 minutes. The SDS forcibly coats all the proteins with a uniform negative charge and completely unfolds them. By eliminating those structural differences, the proteins are forced to race through a gel matrix using an electric current of 80 to 150 volts based entirely on their molecular weight. The tiny proteins zip through the gel incredibly fast, while the larger, bulkier proteins drag behind and move a lot slower. Which brings us to Section 3, transferring and blocking. Okay, so to move our newly sorted proteins from that super fragile gel onto a permanent canvas, we use what's playfully called a wet transfer sandwich, and you literally stack the components in a very specific order. The gel goes right against the membrane, which is usually made of PVDF for high binding capacity or nitrocellulose if you want low background noise. You sandwich that between filter paper and sponges, submerge the whole shebang in a trisglycine and methanol buffer, and blast it with about 100 volts for 60 to 90 minutes. That electric current physically drives the proteins out of the gel and immobilizes them directly onto the membrane in the exact same pattern. But we run into a little problem here. That membrane is extremely sticky. It loves to bind to proteins. If we add our antibody detectors right now, they're going to stick to the entire membrane, completely ruining our snapshot with background noise. So we have to deploy a shield. This step is called blocking. By gently shaking and incubating the membrane in a simple 5% milk solution, and yes, I literally mean milk from a grocery store, or BSA for phosphoproteins, for about an hour at room temperature, we effectively coat all the empty, free binding sites on the membrane.
All right, Section 4, the antibody detectives.
And this brilliantly illustrates where the magic of immunology really comes into play. It relies on this elegant two-step lock-and-key mechanism that makes Western blotting just so incredibly powerful. On the left, we have the primary antibody. Diluted in a blocking buffer, this is incubated with the membrane overnight at 4 degrees Celsius, and it is highly specific. It binds only to your target protein and completely ignores everything else. But here's the catch. We can't actually see the primary antibody. So on the right, we bring in the secondary antibody. We incubate this one for about an hour at room temperature. Its only job is to recognize and bind to the primary antibody. And the absolute secret weapon here is that the secondary antibody carries a detection tag, like HRP, which stands for horseradish peroxidase. Now, I want to pause here for a second to highlight a step that is super easy to overlook, but absolutely vital: the washing step. Between these antibody incubations, you must wash the membrane using a TBST buffer. That's a mix of tris buffered saline and tween 20, typically 3 times for 5 to 10 minutes each. If you don't aggressively wash away the unbound floating antibodies, your final result is just going to be a dark, unreadable mess. Precision washing is literally the secret to a clean, highly readable signal. And finally, Section 5, detection and interpretation. Guys, we have reached the moment of truth. Because our secondary antibody is carrying that HRP tag, we can now trigger a chemiluminescence reaction. When you add a specific ECL, or enhanced chemiluminescence substrate, to the membrane, the HRP enzyme reacts with it, and this reaction literally emits light. You rush the membrane over to an imaging system, capture that emitted light in the dark, and suddenly, kind of like developing a photograph in an old-school darkroom, distinct bands appear precisely where your target protein is located. So the crucial point here is how researchers actually interpret this final output data. You're basically looking at 2 key metrics. First up, band intensity. A darker, thicker band tells you there is a higher protein expression level. Second, band size confirms the molecular weight of the protein. You'll also notice researchers run a control, like beta-actin, which always appears right around 42 kilodaltons. If the beta-actin bands are perfectly even across all your samples, you know for sure your preparation was flawless, and any variance you see in your target protein is a true, genuine biological difference. To summarize the sheer elegance of what we just unpacked, look at this quote derived from our source material. Western blot is more than a technique. It is a molecular verification system, combining chemistry, immunology, and physics to reveal proteins hidden inside complex biological systems. Honestly, it is just a huge testament to human ingenuity that we can reach down into the microscopic world and pull out such definitive visual proof of life's building blocks. Which brings us to the end of our explainer today. It remains one of the most powerful and trusted tools in modern life science for a very good reason. So I'll leave you with this final provocative thought. From discovering new disease biomarkers to validating the next great therapeutic drug, How will you use this powerful tool to decode hidden biological mysteries? Thank you so much for learning with us, and keep exploring.